So thank you everyone for joining us this afternoon for our June webinar for the Cancer Genomics Cloud. I'm really super excited to welcome Dr. Frank Lee as our guest speaker for today's webinar. He is a computational biologist and a postdoctoral fellow at the New York University Grossman School of Medicine, where his research centers on developing cutting-edge computational methods to tackle some of the most complex challenges in biology, I think. Dr. Lee has made significant contributions to the field of cancer immunotherapy and single-cell genomics. He's created widely used tools such as Deep Immuno for predicting T-cell immunogenicity, SNAP for identifying tumor-specific splicing antigens, some other, what is this, Bayes TS, hopefully I'm saying that right, evaluating tumor target safety, and other tools for multimodal single-cell data. So wow, that's very impressive. His work has directly supported the discovery of actionable tumor antigens, as well as the creation of a comprehensive antigen database. So today, Dr. Lee will introduce us to his latest tool, ImmunoVerse, which is a comprehensive computational pipeline designed to analyze tumor RNA-Seq data and identify up to 11 classes of tumor-specific antigens. We're thrilled to have him here and learn more about this powerful resource and how it's accelerating discoveries in immunogenomics. So please, everyone, join me in welcoming Dr. Lee. Thank you for being here today.
All right, thank you so much for the introduction. So let me share my screen. So I know we tested before, but just to confirm, are you seeing the right display? Oh yeah, cool.
Looks great.
So yeah, thanks again for the opportunity to present my work. My name is Frank. I go by Frank. I'm a software engineer by training, actually, and I'm currently a postdoc at NYU School of Medicine. So the topics that I'll be sharing today is about tumor antigen discovery and its implication for developing improved cancer immunotherapy. So a little bit of background, as the title suggests for itself, the goal or what we're interested in is really trying to finding as many as possible actionable tumor-specific antigens across different cancers. As we all know, the conventional CAR T therapy targeting membrane protein like CD19 and CD22 has really transformed the standard of care, particularly for hematological malignancy. But alternatively, cancer cell can also present their vulnerability through the mechanism or pathway called MHC or HLA presentation, such that the protein can also present a part of themselves as a very short peptide. The successful identification of such peptide from, for example, MAGE4, KRAS G12D mutation, has led to FDA-approved therapies such as TCR-T, tumor-infiltrating lymphocytes, or cancer vaccine. And particularly, which I will talk about in the next slide in a minute, our lab, my supervisor, whose previous work has been focusing on developing a new class of CAR T, which are called peptide-centric CAR. The essence of that is really to expand the conventional CAR, which usually target membrane protein, to PHOA, peptide HOA complex as well, such that it really expands the utility of the CAR T therapy. So to go a little bit into that, my PI's previous work focused on neuroblastoma, where we touch down to a particular development-specific target called FOX2B in neuroblastoma, which is extremely tumor-specific. And we identify a short 9-mer peptide, which I show on the lower right, that presented by a common HLA. And the FOX2B PC CAR is just entered the phase 1 clinical trial at CHOP. So this is really a very inspiring story for me as a bioinformatician, because if you imagine just this one little peptide and its impact, both for research and for actually can potentially saving lives. So when I joined the lab, the goal for me as a bioinformatician is, can I really improve the target discovery part? Can we find another way to say that more FOX2B, so potentially more clinical trial can be set up, more cancer patients can actually benefit from such approaches? So then the question is, all right, so we know the peptide HOA is a very important types of target, but how do people normally identify such target? So the Swiss Army knife in this field is a specialized proteomics approach, which are called immunopeptidome. So the essence of that lies in the utilization of the pan-natural monoclonal antibody, such that we can immunoprecipitate. So we pull down all the peptide HOA complex, and then we further get rid of the HOA. So only the eluted peptide will be separated, and then we subject those to the mass spectrometry. But as you can see from these slides, the raw data spit out from the mass spectrometer is basically a bunch of pics or chromatography that nobody can interpret. So the paradigm in this field, or how people actually integrate and make sense of mass spectrometry data, is by actually providing or supply a list of potential protein or peptide that can may be presented, may be there, which we call them a database. Um, but in another way to put that, the caveat of such approach, or sometimes people call it database dependent search, is even your peptide is there that is detected by your immunopeptidome, by your empirical experiments. If this peptide sequence is never in your database, then you will never be able to actually identify this peptide. So as a result, it's very important when you're doing the immunopeptidome or target discovery efforts to construct as comprehensible as possible database or search space for antigen discovery. And what we learned in the probably past decades or 2 decades is we know such tumor-specific peptide PHOA or just PHO in general, they can really derive from a diverse spectra of molecular aberrations. As we know, our human have roughly 20,000 canonical protein coding genes. And another class of aberration that people often use is focusing on somatic mutation because of its clean tumor specificity. But those are really just the tips of iceberg because there's so many different molecular aberrations that are listed here, such as splicing events, tumor-resistant pathogens, circular RNA, or even transposable elements, which there are sporadic reports suggesting that they could actually generate the tumor-specific PHOA. But the challenges right now, where the gaps in this field is there's really no easy-to-use computational solution exists that can jointly consider all types of events that are shown here. And as a result, the scarcity of actionable targets still remain as the major bottleneck for a lot of cancer patients to actually benefit from cancer immunotherapy. So in a nutshell, my work is trying to fill those gaps. And to do so, we developed a computational pipeline we named as ImmunoVerse, which we take the RNA-Seq data from the tumor. The first step is to first comprehensively characterize all the molecular events, different class of molecular aberrations that are listed in my previous slides, including fusion, circular RNA, etc. And all those events will be rigorously compared with a comprehensive normal tissue database because we wanted to make sure that the aberration we selected are tumor specific such that they will be safe target and they won't have any off-target safety issues. And once we have those tumor-specific events, we are able to do an in silico translation to get a potential peptide or referred to as the database that I showed in my previous slides, which we can utilize that to search the immunopeptidome data. We utilize a complementary set of software to really try to boost the sensitivity for identification while also controlling the false discovery. And all the final result will be rendered as web portal for people to toggle around and really make an informed decision. And once we build this pipeline, the second thing that we do is we wanted to apply that in a pan-cancer manner where we collect 21 different tumor, particularly the RNA-Seq and the immunopeptidome data from publicly available repository, where in total we collect more than 7,000 RNA-Seq data and more than 1,700 immunopeptidome data. And this leads us to a relatively complete landscape of pan-cancer atlas of such tumor-specific peptide HOA. And we can, for the first time, really see how different classes of molecular aberrations contribute differently in different cancers. To just give you 2 examples, for example, if we look at a somatic mutation or variant that I highlighted this row, No surprise, the immunologically hot tumor, such as melanoma or lung cancer, harbors the most new antigens compared to the immunologically cold tumors, such as neuroblastoma or mesothelioma, which barely harbor any actionable mutation-driven antigens. On the other hand, that I highlighted for the acute myeloid leukemia, we can see a disproportionately high number of antigens deriving from transposable elements and intron retention, which might be linked to the well-known mutation for the epigenetic enzymes such as DNMT3A or splicing factor mutations. So the main takeaway, or in my following 30 minutes or so, I'm going to share with you 6 very short stories. But I'll just first give an overview of what I want the audience to take away from that. So we identified a self-antigen derived from HLA-CR1, which is a very tumor-specific protein in renal cell carcinoma, and we're developing the peptide-centric CAR against it. And my second story is about alternative splicing antigens deriving from a well-known melanoma-specific antigen called PMEL, which is also known as GP100. And the reason why we're interested in that because we believe that could be a safer target and overcome a very common autoimmune side effect called vitiligo, which I'll talk more in detail. We reported hundreds of new mutation-derived new antigens, both shared and private one, that are actually backed up by the immunopeptidome evidence. The fourth story is about transposable elements, where a previously elusive WRF2 protein from a particular type of transposable element called line1, we argue that they may actually serve as a very excellent pan-cancer targets. And last but not least, we're not the first group reporting that, but we also find really a significant contribution from a cryptic open reading frame, which I'll talk about in a bit as well. And also there are enormous amounts of pathogen-derived antigen from virus, from bacteria, that might also be promising target for therapeutic cancer vaccine. And I'll end my talk with a very short description of the tool and web portal that we built that are publicly available for the antigen discovery. So let's get started. The first story is about HLA-CR1. This gene is also known as KEAM1 or TEAM1. The HLA-CR1 is a known oncogene that really drives the renal cell carcinoma tumorigenesis. Although this gene itself is actually a membrane protein, but the actal domain shedding really hinders the development of the antibody using conventional CAR T because you never know what is the cleavage site of this protein. So alternatively, through our analysis, we actually found this HLA-CR1 protein manifests itself through the HLA presentation pathway, as I illustrated in my background slides. So a very short epitope from this protein that I show on my right-hand side can be presented by a relatively common HLA allele called B08, which covers around 18% of US population. And from the computational modeling that you're seeing from here, they actually created a very interesting surface area where they have two charged residues protruding to your receptor. So that makes developing a potential binder way easier compared to, for example, hydrophobic surface. And as I mentioned, the HLA-CR1 is a very, very tumor-specific gene by itself from the RNA level, which I show on the left. But more importantly, because the actual target we're talking about is this peptide. As you can see, this peptide is also super, super abundant, and more importantly, it's recurrent detected across many different tumor samples and are super clean in the normal tissues, which render them a very excellent targets. So we're not developing PC-CAR against it. There are data that we have been generated, but we're now doing more orthogonal validations to make sure the killing and the functional assay that we have done are actually the target specific. So that's the first story. The second story is about the a melanoma target, the spliced variants of PMEL with GP100. So a little bit background or motivation about this story is vitiligo is a very common autoimmune side effect for current melanoma immunotherapy. So as you can see on the left, although the vitiligo itself is a relatively harmless side effect, but it indeed reduces the quality of life for people that undergo the immunotherapy. And the reason why it is a commonly observed side effect for melanoma immunotherapy is mainly because the anti-melanoma immunity usually is mediated by a class of differentiation antigen that are often also expressed in the normal melanocytes or skin as well. So as you can see, one of the examples is this gene called PMEL, also known as GP100. Although people often say that's a melanoma-specific gene, but if we really zoom into its normal tissue expression, what you can really appreciate, which I highlighted in this red box, is on the skin tissue, there are appreciable level of expression, which explains the depigmentation that underlie the common side effect that I'm showing here. So the motivation for us is really trying to figure out whether we can find antigen from PMEL that are potentially safer, that can potentially, overcome the common side effect while not compromising the therapeutic potency as well. So what we found is actually a splice variant, which I show on the left. So normally the PMEL has the 3 exon 4, 5, 6. But in the meantime, in the melanoma cells, they sometimes often skip this exon 5, which is shown here. And because of this novel junction, they actually create spliced variants from exon 4 direct to exon 6. And although the peptide seems to be similar, but if you really look at that carefully, you can see they have very, very different peptide sequence. First of all, this splicing junction is extremely tumor specific. And then based on the computational modeling that I'm showing on the right, I hope you can also appreciate that from a structure perspective, there are very different sterical conformation for the splicing variant versus the what we call canonical one, or the one that actually used in clinics. So the reason why I think it's maybe a safer target is when we really look at the expression profile or therapeutic window between this splicing junction versus the canonical one that I use clinics, where we compare the expression in melanoma and the sun-exposed tissue, which has appreciable level of melanocyte. Here, I use the 3 red boxes to really do a side-by-side comparison. As you can see, the canonical junction, which gave rise to the antigen that used in clinics right now, has a significant amount of junction rate or expression. Remember here, the y-axis is in the log scale. But on the other hand side, you can see this novel splicing junction has a super, super clean, almost zero expression in the normal skin, which is very important because that's exactly the goal that we're trying to achieve to overcome the potential autoimmune side effect. And then the second question we're asking is, okay, you convinced me that they might be a safer target, but would that compromise the therapeutic efficacy, how potent they could be? So further, through our immunopeptidome analysis, we wanted to quantify the abundance or the intensity between our newly discovered splicing antigens versus all the canonical antigen. And as you can see from this slide, very surprisingly, our splicing antigen stands out as the most abundant one and even statistically significant than some of the canonical antigens. And the reason for that is actually not completely unexpected, because we know there's something called, so all the unstable spliced product are normally prone to be degraded very quickly. And because the actual presentation pathway also require the protein to be degraded first, so this thing actually rendered them more favorable to be presented. There's a dedicated hypothesis called DRIP, stands for defective ribosomal hypothesis, that specifically explains why such phenomena could happen. So in summary, through our computational analysis for the second story, we find the spliced PMEL variants that actually can potentially provide a safer and more abundant antigens compared to the canonically peptide using currently in clinics. So the third story I wanted to share today is about neoantigen or mutation-driven antigens that are widely used in clinics for therapies such as cancer vaccine and the tumor-infiltrated lymphocytes. So something that people may not be aware of is currently to meet the fast turnaround time for cancer vaccine, the standard of care actually heavily relies on the computational prediction, despite of the fact that the actual proportion of such predicted binder are as low as 2%. So the hypothesis that we are really trying to pursue here is we argue the new antigens that are backed up by both the computational pipeline and the immunopeptidome may actually have higher confidence and should be prioritized for vaccine and TOS expansion because they're guaranteed to be presented, they're more high confidence, and probably also more high abundance as well. So now we have this atlas. The question we really wanted to ask is, can we do a comparison? So we took all the mutation-derived antigen from our database, and we compare with a previously published database that completely based on computational prediction. And as you can see on the left, the construct is actually super daunting. The intersection only account for less than 0.001% of all the predicted antigen. Although we think this number may be underestimated because of the sample size, actual restrictions, but they really emphasize how important it is to actually account for the immunopeptidome evidence as well. But on the bright side, I think we can also appreciate there's 471 antigens in the middle actually represents very promising antigens because they have the evidence from both sides. And if we really zoom in, the most frequent one is from colon cancer, particularly a frameshift mutation from this gene called LARP4B, which likely due to the microsatellite instability in the colon cancer. And there's so many different shared mutations that are listed in this table. Some of them are known, some of them are novel, which we believe should gain some attention when we're considering your antigen cocktail or for a TOS expansion. So now we know that this atlas is very helpful for us to prioritize such shared antigens. The next question we want to ask is, because we know such new antigen a lot of times are not shared, they are presenting a private manner, meaning that usually it's personalized. So we want to ask the question whether ImmunoVerse, the atlas that we're building, can actually also be utilized in a personalized medicine. So to just give you a background, like I showed before, for the conventional cancer vaccine development, usually we take the cancer patient's WGS data, the genome sequencing data from tumor lesion and match normal, and we also incorporating the RNA expression data to really get down to a list of expressed somatic mutations. And then we do the computational modeling, predicting the binding, and then prioritize, like say 20, 25 top hits, and then reinfuse them back to the patients. So what we are proposing here is a very, very simple add-on. When you have those expressed somatic mutations, before you do all the things here, you can just do a very simple search using our publicly curated immunopeptidome. And we also incorporated a feature for you to immediately assess the confidence of such identification, such that you can then go back to your conventional workflow. So it's completely integrable without any extra work. So to actually demonstrate our proposed workflow. So we take one melanoma patient where we have the WGS, tumor, normal, and tumor data, where we narrow down to 534 expressed somatic mutations, and then we search against 267 public melanoma immunopeptidome data set that we curated from our study. Through this process, we identify 8 such neoantigens that can actually be evidence from our publicly immunopeptidome data, and then our pipeline is able to using the in silico spectral validation to readily help us to assess what's the confidence of this identification. In this case, I show one of the case, which we believe are of high confidence. And then we go back to, among these 8 antigens, now we go back to the normal workflow where we predicted the binding affinity, immunogenicity. So through this manner that we can really narrowed down to a list of antigen that can potentially be more immunogenic, higher abundance, and have evidence from both sides for us to justify their inclusion for cancer vaccine. And most importantly, the whole process takes less than 4 hours. So it's really not a burden, which we believe that can be integrated into virtually all the conventional cancer vaccine that currently used right now. So that concludes the third story. So to switch gear a little bit, the fourth story I want to share is about transposable elements. So transposable elements is a very interesting types of elements in the genome. Sometimes people call them junk DNA or jumping genes. And particularly line 1, which stands for long interspersed element 1, is the major driver of mobile DNA activities in modern human genome. And particularly this line 1 element encode 2 very important proteins that help them jumping around our chromosome, which term WRF1 and WRF2. And these 2 proteins have to cooperate with each other to actually coordinate or enable such transposon activity. But mysteriously, through all those years, scientists actually failed to detect the native WRF2 protein in a lot of conventional experimental approaches, including bulk proteome, immunohistochemistry, and even Western blot. So the hypothesis we're trying to pursue here is maybe the WRF2 protein is fast degraded. That's why they never be able to detect it through the conventional approaches. But as I alluded to in my splicing story, because fast degradation is actually a prerequisite and make them more prone for HLA presentation. So we're arguing maybe through our immunopeptidome analysis, we're able to find some clue of this protein. And as expected, through our pan-cancer immunopeptidome search, we're able to find a lot of fragments, peptide fragments from this WRF2 canonical protein, which are mapped to the and shown on the right. And for some fragments, they are recurrently detected in more than up to 8 different cancer types. And then to really demonstrate that they might be able to use in therapeutic setting, we further test how tumor-specific they are. And to do that, we not only just search for those peptides through the cancer immunopeptidome, we also further search their existence in a normal immunopeptidome as well. And here you can see there's this extremely clean therapeutic window for the WRF2 protein. They are almost completely absent in normal tissue. And for some of them, like I said, they are pan-cancer, they are detected across different cancer, which render them as potential pan-cancer targets for a lot of cancer that are currently lack actionable antigens. So now we're so after the transposable elements, the last 2 story is not new, but it's also super cool that that we can recapitulate what had been found before in this field. So microprotein, which also sometimes people refer to them as a cryptic open reading frame, is normally refers to the types of open reading frame that are different from the annotated one, which are shown on the left. They can be residing on the 3' upstream region, or overlapping with the annotated one, or slided out of frame, or all the way to the 3' downstream regions. So through our analysis, we're able to find a bunch of such antigens deriving from different regions that are likely tumor associated. And to further confirm that, because of their cryptic, so to really prove that those peptides are translatable, we wanted to be sure and they are actively translated region. So we conducted RiboSeq for 8 neuroblastoma PDX model, where we have the immunopeptidome data, and we also further conducted the RiboSeq for exact match tissue. And through our analysis, we found for some of them, they are actually active translated that are supported by the ribosomal occupancy for that particular peptide region, like I shown on the panel C, the lower half, where you can see the highlighted peptide that particular open reading frame are robustly evidenced from at least 4 ribosomes that are literally indicating that this is a correct open reading frames, which give us more confidence that this is a true identifications. So last but not least, the tumor-resident pathogen is always a very interesting topic for a lot of people working in this field. As we know, the viral infection, such as HPV or HBV, has been linked to cervical cancer or liver cancer for a long time with very well-studied mechanisms. And also, we know that the preventative vaccine for such as HPV vaccine, HBV vaccine can drastically reduce the incidence of such cancers. And the thing that we also know in this field is from a therapeutic perspective, since the HPV, HBV are present in the cancer lesion, so such virus can actually present part of themselves through the cancer cells actually HLA molecule. And through our analysis, we're able to detect such antigens that directly from the virus proteome in, for example, the HPV antigens in cervical cancer and head and neck cancer, and also the HBV antigen from liver cancer, for example. We're also able to find other virus evidence, for example, the EBV virus in head and neck cancer. And surprisingly, we also found some evidence for the CMV in cancer such as neuroblastoma and ovarian cancer that we are currently validating. And on another hand, there are also some well-known cancer-associated bacteria such as H. pylori for stomach cancer, and also Fusobacteria nucleatum, which is a known bugs associated with colon cancer and melanoma. We are also able to find some evidence that those pathogens can actually utilize the cancer cells HLA presentation pathway to present part of themselves and then be recognized by the immune system. And the thing that I show so far are all known biology. There's nothing new. But actually, through our analysis, we're able to find a very interesting bug, which is called Nealia circulans. What we found very interesting is this particular strain are detected in more than 90% of ovarian patients. And to further make sure that what we're seeing here is not just because of some batch effect or some technical issues, we collected all the RNA reads that we know are mapped to this particular strain from different institutions. And you can see the RNA reads can be uniformly mapped with 100% identity across the whole bacteria genome or transcriptome. And more importantly, on the protein level, it also have a very, very wide coverage for a lot of protein coding genes. So those really give us more confidence that they might be true. And now we're also conducting more orthogonal validation to further figure out that what's the function of this bug, why they're here, and what can we learn from that. And last but not least, I wanted to give a shout out to the Seven Bridges platform, the Cancer Genome Cloud, which really helps a lot with this whole project. And besides all the biological insights that are presented so far, we are also releasing a very interesting and potentially useful platform, which we call them a code-free. Means you don't need to write a single line of code, and you can do all the things that are presented so far. When you have RNA-Seq data, you can just profile all different types of molecular aberrations in a code-free platform. So to just give an example, let's say you have some RNA-Seq data, and you're really interested to ask the question, oh, how does the splicing and internal retention profile look like in my samples? What you need to do is to just input your raw sequencing data, and then click the run, and then you can get the results after 10 or 20 minutes. So which is super helpful considering without dealing with all the nitty-gritties for how to set up the dependency, how to run the environment, and you can just get a result and immediately interpret the data. And then we also develop an interactive web portal for target selection. And I will try to give a live demo for that. So I'll just first stop sharing and then see if I can go to the web portal. Let me know if you're not seeing the right window, but right now I am at the URL that I show here, called immunoverse.com. So the reason why we developed this web portal is really try to democratize antigen discovery efforts. We know for clinician, a lot of time they really wanted to have a way to visualize all different properties associated with all the antigens and try to make an informed decision in terms of which one to go for experimental validation or even go forward. So here you can select different cancers, or you can select all. In this case, if we select a neuroblastoma, you can select different types of antigens or all antigen if we don't want to be specific. For example, if we wanted to see the one that I showed before right now, it's currently entering the clinical trial. It's from Fox2B, and we go submit. You can immediately see this particular peptide right here. And if you just click this peptide sequence, you are able to see how tumor-specific this gene is, which is probably the most important thing that you should consider when you're developing a therapy against. And then, because the mass spectrometry sometimes is inherently suffer from false discovery, for people who have the expertise to actually interpret the peak results, we actually show the raw identifications for this peptide. In this case, is a super confidence identification. This can give us extra credibility, say that, oh, this is a very good target. And then when we look at the peptide intensity, we can see this peptide is super, super highly recurrently detected across different HLAs. And they're, of course, also ranked as very high in terms of the percentile. You can also see the ranking plot from on the right as a more granular view of this peptide. And here you can see all the HLA that can potentially present in this peptide. What's the frequency that it can cover for the US population, and how many sample actually this peptide get detected, and what's the recurrency, and potentially what's the immunogenicity if you are wanted to go for cancer vaccine or TCR-T therapies. So we really hope that by utilizing this interactive web portal, that the clinician can really make a decision themselves and not need to go back and forth, working with bioinformatician to figure out that which one has or can check all the boxes for us to really make decisions. Say, oh, this is a good target to move forward or not. So let me stop sharing for that part and then go back to my slides. So with that, I really wanted to, I don't want to go this one because I've already showed you sorry. So let's just go back to the acknowledgement slide because I really want to thank my lab and a lot of collaborators here at NYU and across the country. And of course, like I said, big shout out to Seven Bridges, Ro and Sarah, and many people that have helped me a lot with this project. They're super helpful and highly recommend working with them for sure. And with that, I'll stop and I'll take any questions.
I wanted to thank Dr. Lee for your presentation and for sharing your work with us. Your research and tools are clearly making such a meaningful impact in cancer and immunotherapy and beyond even. So thank you to everyone else who joined us today, actually. So I hope you found the session valuable and inspiring. If you have any follow-up questions or you'd like to learn more, please don't hesitate to reach out to us. I'll be sharing a recording of the webinar shortly, and as well as relevant links and resources and things like that. You can find everything on cancergenomicscloud.org/webinars. So thank you again. We look forward to seeing you for a future session.
Cool. All right. Thank you so much.
Thank you.